Good afternoon, everyone. Our keynote speaker today is Dr. Lucia Peixoto. Good afternoon, everyone. Our keynote speaker today is Dr. Lucia Peixoto from the University of Washington at Pullman. Dr. Peixoto, Dr. Fisher, if the pronunciation sticks in your craw, has a lengthy resume of accomplishments, not the least of which is her recent election to the board of directors of the International Society for Computational Biology. Dr. Peixoto studies neurological development and autism, and will be presenting today on reproducible research in neurobiology and informatics.
Thanks, Tim. I'm going to share my screen now, so bear with me. Can you see the screen? Everybody good? Thank you. I first want to thank the organizers for inviting me. This is truly an honor. I've been a BioC user since my early days as a graduate student, so I am really excited to be able to tell you a little bit more about my work. So first, I want to start telling you what my lab does. And our goal is to understand how sleep and learning are altered in autism spectrum disorders. And we have quite an emphasis on transcriptional regulation. So we do this mostly in mice. We combine mouse behavior, meaning that we just make the mice do things. We do recordings of neuronal activity, and then we collect a lot of transcriptomic and epigenomic data sets, which means that we have to analyze the data, and we want to make sure that what we find is reproducible. So today, I'm going to talk to you about a journey that I started about 10 years ago on trying to understand what the effects of experience are on transcriptional regulation in the brain using omic approaches. And the two experiences I'm going to tell you about are learning and sleep, and mostly sleep deprivation. And as you probably have some personal experience, sleep deprivation and learning interact with each other. But before I tell you about my data, I wanted to tell you about the journey and the challenges that we face, and a little bit of what we learn about how to overcome those challenges. So there's some important considerations for overcoming the challenges that we face, and this may seem obvious, but my experiences, they not always are. Good experimental design is crucial to be able to answer these questions, and this means that you have to know your variables and have controls. And one of the things that is really atypical about the brain is that when you introduce a variable, you're always introduce it in a sea of many variables. The brain in vivo is never off. So it's really important that before we move on to understand a typical or a disease or pharmacology, we first understand what is typical. Otherwise, if we introduce those variables at the same time, it may be really hard to tease them apart. The other thing that I have learned over the last 10 years is that biology is biology regardless of technology, which means that as soon as a new technology is introduced, that should not erase the knowledge that we have gained from previous technologies. So today, I'm actually going to walk you through how we have looked into using different technologies like microarray, RNA-seq, and most recently single nuclear RNA-seq to look at exactly the same problem. And you should see some level of reproducibility between technologies. And if you don't, then you have to ask yourself why. So one of the challenges on collecting omic data and the experimental design is that omic experiments usually measure thousands of events simultaneously. So a good experimental design will minimize the effects of things that you don't care about it. I call this confounders. Some other people call them batch effects. So from now on, I will say confounders or confounding variables. When you design your experiment, you ideally have controls that will allow you to estimate the effect of the treatment versus the confounders. And when you cannot control something, you need to try to randomize it. There are many, many variables in omic experiments, both technical and biological, that can affect your measurement. And if you don't account for them, you may not be able to detect the signal of what you're interested in. So confounders are always a problem. So the real question for any specific experiment is how much of a problem they are. And the confounders are present regardless of technology. So I sometimes have discussions of moving on to a technology when microarrays were replaced by RNA-seq, people ignoring the confounders that we knew existed before. one of the things that I learned through hard work is that in the brain in vivo, biological confounders tend to dominate the signal of interest and sometimes are unknown or hard to control for. So when you initially get the data doesn't make any sense. But eventually, if you're analyzing it properly, it will. So how can we account for the effect of confounders? The step in the data analysis that accounts for that is called normalization. Many of you are probably familiar with normalization of data sets. And it's really important that you explore your data to try to understand what the confounders are, and also explore the data again to see if you actually remove them. So how do you know if you have removed the confounders in your data? Ideally, if you have replicates, they should be more similar to each other, and they are two samples from different conditions. The samples after being normalized should have comparable dynamic ranges. The p-value distributions should be mostly uniform. And then the last question of them all is, do you have controls, and how are your controls performing? And I often talk about there is no one-size-fits-all pipeline. There are certain steps that are common to all analysis, but the contribution of confounders is unique to every data set. So your normalization is probably going to be different in different scenarios. So I'm first going to start talking about a work that I started as a postdoctoral fellow in the laboratory of Dr. Tadable, trying to understand the effects of learning in genome-wide gene expression and epigenetic regulation. So first, I'm going to just exclusively talk about gene expression. And why are we looking at gene expression? We know that long-term memory formation requires transcription. So after you make a mouse learn a task, and I'm going to tell you a little bit about what we make them learn in a second, about 30 minutes or so after that task, if you pharmacologically inhibit transcription, the mouse will not remember what it learned. So we know it's important. There had been a lot of studies trying to use omic approaches to understand the transcription of which genes matters. And at the time I started this work, there was very little agreement between studies, and the studies had very little power. For example, people will forego multiple testing correction, because if you did multiple testing correction, you won't find any genes. And you can probably agree with me that is not a very good idea. So what we learn in the process is that what you really need to actually answer this question is proper normalization. And proper normalization then increases both the power and the agreement across studies. So what do we teach the mice? We use a task called contextual fear conditioning, and it works like this. You place the mouse in a box. They've never been in that box before. So the mouse explores the box, so it's learning the spatial navigation, the shape of the box. And after 5 minutes of exploring the box, the mouse is shocked in the paw. That is a very classic sort of association task. We then place the mouse in their home cage, and the following day, if the mouse remembers that it got shocked, when you place them back in the box, it will retrieve that memory and it will freeze. It will try not to move, so it won't get shocked. So in order to collect data for this, to look at gene expression, we did the following design. So we started our fear conditioning at 10 a.m. one day. We have to have controls for time of day, because time of day is a very important regulator of gene expression in the brain. And in the first time point that we took was those 30 minutes right after that we knew were important. And then we took a time series, 4 hours, 12 hours, and 24 hours afterwards. And then we also wanted to know, when you retrieve the memory, how similar is the transcriptional profile to the profile that you have when you learned it. So this is the design that we used, and we repeated this experiment every day for 9 days, so we can have 9 replicates, independent biological replicates per time point. And this was a microarray study that we did. And initially, I started working with Dr. Nancy Zhang at the University of Pennsylvania, and I thought I had a really clever experimental design and was going to be able to answer the question. And when we look at the data, that was not the case. So what I'm showing you here is the 3 principal components, and I'm showing the loadings, because if you actually do a 2-dimensional representation, it's too messy to explain. So color-coded at each of our sample groups, and we have 9 replicates in each of them. And what you can see, let me see if you can see my laser pointer. Here we have the controls and the fear conditioning. We expect a pretty big effect size here, between these two. And the first two principal components will not capture the effect of the treatment. Only in the third principal component, and by the way, combine this, we're over 50% of the variance in the variability in the study. Only in the third principal component, we can start see a sort of the effect of the treatment, but we also saw another effect overlay with that, and I hope you can follow my pointer. And this is circadian gene expression. So we knew at this point that we needed somehow to account for this variability if we wanted to see anything that was due to learning. So what we decided to do with Dr. Zhang was to remove the first principal component completely. And after we did that, we actually were able to obtain a reasonable or at least expected clustering of the samples and a certain number of differentially expressed genes. Without removing the first principal component, you don't see any differential expression at all, at least not surviving multiple testing. So what we saw here, this is the number of differentially expressed genes between controls and either 30 minutes, 4 hours, or 12 hours after learning, is that there's no overlap at all between these three data sets, but there's quite a bit of an overlap between the learning and the retrieval of the memory. So we went on to validate what those processes are. If anybody's interested, that is a BMC genomic paper of 2015. But one of the things that we learned from here, and if you look at the number of genes that are differentially expressed, is that we don't have a lot of sensitivity. So by then, RNA-seq had come along, so we decided to repeat this experiment and use RNA-seq instead. Now we're going to be focusing only on the 30-minute time point, which is where we had our strongest effect size. So our RNA-seq dataset has 5 biological replicates, each of either the fear condition in the control or the retrieval that happens the following day, exactly at the same time of day. But now we have the microarray data, which is completely independent, which gives us both negative and positive control genes to test how well we're doing. And we're going to test the effect of different normalization methods for RNA-seq data analysis. Most of the traditional ones, such as upper quantile, TMM, or PKM, only correct for library size. And at the time, and after looking at what the problems that we had with our microarray data, I contacted and a professor I met at a conference. His name was Terry Speed, and he told me his postdoc, David Ariso, was developing a method that will probably be better at accounting for the level of noise or confounders that we had in the data. That is, remove unwanted variation for sequencing, or RUV-seq, if you're interested in looking into that method. David Ariso developed it and is published in Nature Biotech in 2014. So long story short, indeed, RUVS or RUV is the only method that allows us to isolate the effect of learning. So what I'm showing you here are the relative log expression plots and the PCA plots of our data, either upper quantile normalized or using RUV, a version of RUV called RUVS, in which we are using both negative control genes and samples, meaning the biological replicas, to try to remove the unwanted variation in the data. You can see the dynamic range of the samples is much more comparable, that we get a separation with the controls of the treatments. And the question is, do we get any difference in the identification of differentially expressed genes? And we do. So this is a volcano plot of log fold change over p-value. And now what we have color-coded in red in here are the positive controls for our microarray study, which is independent. And in green, we have the negative controls for the same microarray study, which we define as genes that had a p-value greater than 0.8. And with upper quantile, we are only able, and an FDR 0.01, only able to detect 32 genes being upregulated and 2 downregulated. We retrieve about 60% of the positive controls. And when we do pathway enrichment, we only find that the MAPK pathway is enriched. And this is a pathway that we know is regulated by learning. However, after our UVS normalization, we obviously increased the power, we increased the detection of positive controls, but more importantly, now we're retrieving a lot of pathways that we also know have an established role in learning and memory. This was published in Nucleic Acid Research in 2015, so this is quite old. You're welcome to take a look. The other thing that we notice is that our UV increases the concordance across platforms. So what we have here is the genes that are ranked by lower to higher p-value, and whether there is a concordance between RNA-seq analyzed with upper quantile and the microarray analyzed by Lima without any RUV correction. And here we are just changing the normalization of the RNA-seq data. And you can see that RUV, only doing RUV on RNA-seq about doubles the concordance between the two platforms. So just to summarize what I told you about, RUV seems to be deals with the confounders that are left untouched by all the other methods. We know this because it gives better results that actually match the biology, know it exists. We don't need to know what the confounders are, but what you really need to have is biological replicates and ideally also controlled genes. So after we looked at gene expression, I wanted to look at the effects of learning in chromatin accessibility. Why? Because we know that epigenetic regulation is also necessary for long-term memory formation. And I'm going to show you in a little bit that I initially did an experiment that showed that learning has a large overall effect in chromatin accessibility. But when I was using all the standard pipelines available at that time to find which genomic regions were differentially accessible, I could not find any significant effect of learning. And that puzzled me. So which experiment we did? We did the same fear conditioning task as I showed you before. These samples were collected at the same time as the RNA-seq samples I showed you a minute ago. We are taking the hippocampus, which is an area that is heavily involved in learning and memory, the same area of the brain that I use for RNA-seq studies. And we use something called SonoSeq, even though it's not a very popular way to look at a chromatin accessibility. It is a published way. And at that time, this was before ATAC-seq was commonly used. We have 4 replicates per condition. And what I'm showing you here is if you basically aggregate the signal over all the transcriptional start sites in the genome, and what we have here is the average over the 4 replicates, you can see that the fear conditioning samples have a pretty large effect on the opening of chromatin accessibility. But one of the challenges that we had at the time is that epigenomic data, unlike gene expression data, when you were looking at gene expression data, you know where the data is going to map on the genome. It maps to the genes. Well, in epigenetic data, you first have to find the regions of the genome that contain the signal, which is called peak calling. And one of the things that we noticed right away, and this is something that I was working also with Dr. Zhang, is that the peak calling is often not very reproducible across replicates. And we also had the problems with exactly the same confounders that we saw on our RNA-seq data were also present in this data because it was the same experiment, or at least the same experimental setup. So how did we go about trying to solve this? We developed something called the SCAN, Differential Enrichment Scan for Epigenomic Studies. When I started my lab, a student of mine, John Koberstein, continued to work on it. And DSCAN is a R-based pipeline for the differential analysis of epigenetic data, designed mostly to deal with broad signals such as chromatin accessibility or histone modifications. So the first stage is a peak calling stage that is done in R. So we adapted an algorithm that Nancy had developed for scanning through the genome that uses an adaptive window size scanning to call peaks without the existence of previous input. And then what we do is align the peaks across replicates to select what we call the confident regions. So what you are seeing here is the performance across 4 replicates of the same condition. In this case is the control. And how many peaks do you have if you require 2 replicates to agree versus 3 replicates to agree versus all 4 replicates to agree. And the way we use this, although this can be changed or tailored to what you're doing, is we require all replicates to agree, and we call this the confident peaks. And with this, you assemble a count rate matrix that now looks very similar to an RNA-seq count matrix, and you normalize it using RUV the same way that we did in our RNA-seq data. So when we actually do that, now here what you have is the performance on differential accessible regions between the comparison between using upper quantile or RUV normalization, the PCA plot, the p-value distributions, and the volcano plots. And without using RUV normalization, even though we're using the reproducible peaks, you don't detect any differentially accessible regions. Well, when you actually, after RUV normalization, you detect about 2,000 or so. We went on to actually show that those differentially accessible regions, after learning, are developmentally regulated, they are bivalent enhancers, and they are linked to the expression of genes, not at the fear conditioning state, but after the retrieval. And they're associated with autism genes. And we published that in Science Signaling in 2018. So how does now Dario Righelli from Davide Rizzo's lab then took the task of turning DSCAN into a bioconductor package. Is called DSCAN2. And what I'm showing you here is how DSCAN2 compares to other approaches for differential chromatin accessibility. So in here, we are using a data set that we got from this paper, which is the neural activity effect on chromatin accessibility. And here by neuronal activity, they're just shocking the mice, is the equivalent of electroconvulsive therapy. This is ataxic data. There's 3 biological replicates per condition. And what we are comparing here is either DSCAN, which does both peak calling and consensus matrix and differential enrichment through HR. The pipeline that we see most often published, which is doing the peak calling with MAX2 and feeding into diffbind, which is a bioconductor package, or Seesaw. So one caveat of this is we really don't know what truth is. So we're just comparing how they perform relative to each other. So what you're seeing here is an upset plot, or either running DSCAN with default parameters, diffbind with max using the narrow peak call, Seesaw, or diffbind using max with broad peak parameters. As you can see, these are the number of differential accessible regions that all of the approaches agree with. And DSCAN2 actually has the lowest number of differential accessible regions that are unique to its to the method. So it is our thought that DSCAN seems to have a lower false positive rate than other approaches. But I would love if people give it a try and you tell us what you think. So as a summary from part 1.2, DSCAN integrates peak calling across replicates to produce reproducible peak locations. By default, it requires biological replicates, at least 3. I think replicates are important. Additional normalization is still necessary because there's usually additional confounders besides the peak location. But I hope I convinced you that our biological conclusions, at least after learning, go from finding nothing to identifying learning regulated regions that have important biological signal. DSCAN is now available in BioC, and here is the OI for you to find it. So in the second part of my talk, I am going to talk about the effects of sleep deprivation in the brain. And the reason that we are interested in this is because there's been several studies showing that sleep deprivation causes a substantial changes in gene expression in the brain. And I also started this work as a postdoctoral fellow because we also knew that sleep deprivation inhibits learning and memory formation. And at that time, again, multiple studies had been done genome-wide, but they didn't agree very much. And when I joined the lab, a graduate student in the lab had collected a microarray study of sleep deprivation, and that had done about 60 qPCRs, and the concordance between those 2 data sets was very, very small. So Ted, my advisor, asked me to look into it. And again, I'm going to sound like a broken record here, but we learned that what we really needed to do is normalize the data properly. After you do that, you're going to see that sleep deprivation has a very large effect size. So it's probably, for all the stimulus I've ever done in the context of neuroscience, the one that has the largest effect size. So think about that next time you stay awake for too long. And then we can improve the agreement between studies, which was our goal. So how do we do sleep deprivation? And as this is actually a very important issue, because the way you keep a mouse awake can have very different effects on the molecular biology downstream. Why? Because sleep deprivation is stressful. So a quintessential question in the field is it sleep or is it stress? A lot of automated ways of sleep depriving animals involve procedures that by default are going to elicit a very strong stress response. So the most commonly used one is to put the mouse in a little pan with water. So when it falls asleep, it basically gets waterboarded. Or to have a spinning rod that basically hits the mouse in the face every time it tries to fall asleep. And then in those contexts, any molecular response is going to be mostly the response of stress. So we do something called gentle handling, in which we keep the mice in their home cages, and we use something very technological advanced called a paintbrush. And every time the mouse tries to stay awake, we just tickle it with a paintbrush. And we do that for 5 to 6 hours in a row. And after that, we can also then let them fall asleep and see how different naps can reverse that effect. So it's a very good system. We can reverse this very easily. So as I told you before, when I started in my lab, in my postdoctoral lab, neuroscience lab, somebody has done this experiment using microarrays. The other important thing is that you always have to have time of day match controls. I'm going to go through a long story really shortly, but if you're interested in this, you can look into these references. I took the data set. I realized the normalization was not done appropriately. I fixed the normalization step. Now we had 100% retrieval of the independent qPCR data set. And all of a sudden, we saw something in the data that nobody had ever seen before, which was the downregulation of eukaryotic initiation factors, or EIFs. When I showed this data to my mentor, he told me that this could not be true, because this implicated that sleep deprivation potentially will inhibit protein synthesis. So we went along and we looked at signaling through the mTOR pathway, which is a key pathway that regulates protein synthesis. And lo and behold, sleep deprivation downregulated mTOR signaling. Jen, which was another postdoc in the lab, then went along and did a global protein synthesis assay. And again, sleep deprivation did downregulate protein synthesis. So we validated this finding. And what she did, which was really nice, it then injected eukaryotic initiation factors in the hippocampus of mice. And he showed that by doing that, you rescue the effect of sleep deprivation in learning and memory. So when I started my lab, I wanted to see if we could tackle the issue of lack of reproducibility across labs. And we move on at looking at a different part of the brain called the prefrontal cortex. The hippocampus is really well known for being involved in learning and memory, but people that do sleep usually look at the cortex because the cortex is more involved in executive function, and higher order function is heavily affected by sleep. So what we have here is microarray data, and the different shapes are different labs. And in orange, we have the controls, and in green, we have the sleep-deprived samples. So when we try to normalize the data together using RMA, obviously, the biggest source of variability is the lab. Then I went on and I looked into my hippocampus microarray data, and I also asked how many of the findings in the hippocampus had been replicated in the cortex. And I assembled a data set of about 25 positive controls, which are these genes in red, so that we can actually benchmark against them. Now, if instead of using RMA, we use RUV to normalize this data set, now we can get rid of the interlab effect, and we can recover about 94% of positive controls. And all of a sudden, now you can see all these genes in blue, which are the genes that are differentially expressed after sleep deprivation based on microarray across 3 different labs. And these genes are going to be important because now they're going to become our positive controls for our next set of experiments. So we wanted to improve our understanding of transcription regulation after sleep deprivation. This is our experimental outline. We have animals that are either allowed to sleep or they are sleep deprived by gentle handling for 5 to 6 hours. We take their brains and we either take the bulk tissue and isolate RNA, or we do nuclear isolation for single nuclear RNA-seq. We are doing this in wild-type mice. So far, I've been telling you everything about typical, but we are doing this also with our autism mouse model, which is a model that has a mutation in a gene called SHANK3. So we did the bulk experiment first. We did Illumina sequencing. We have 5 replicates. We're sequencing pretty deeply. And in parallel, we did 10x V3 chemistry with Illumina sequencing. In this case, we have 3 replicates. And the reason that we have 3, and I'm going to show you this in a moment, is because sleep deprivation has a very large effect size. So I felt comfortable with the replicates, about 10,000 nuclei and 30 to 50,000 reads per nuclei, meaning that this to have comparable. Can people hear me? Comparable read depths. Just in brief, I'm going to go to the data analysis outline, and this is mostly as a reference, right? So our bulk RNA-seq data analysis outline is pretty much set, while our single nuclear RNA-seq is a work in progress, but is so far, this is what we're doing. So we quantify reads using Salmon, either Salmon Quant or Salmon Alanine. And for our bulk RNA-seq, we do QC with AdaSeq, we normalize with RUV-seq, and differential expression analysis using HR. When we are doing our single nuclear RNA-seq, and when we're looking at the brain, and differential expression in the brain is really important to do nuclei and not cells, and I'll be happy to take questions about it for anybody that is interested. But the bottom line is that in order to dissociate neurons, neurons, when you dissociate them, will send a very strong transcriptional signal that will obscure the effect of your treatment. So you have to actually take the samples first and dissociate the nucleus, and that works better. Because of that, we need to do a data setting processing step, because the reads, the RNA you get, the reads are not processed, so you need to include the intros when you do this. And this is our current pipeline. So QC with MyQC, we do normalization and back correction with Scranton Bachelor, and we ended up going for cell annotation using our reference data set. I'm going to tell you a little bit about why in a second. And then we are faced with a problem of differential state analysis, right? So we have a multi-group or multi-population. We have all this population in a single nuclear RNA-seq, multi-sample kind of design. And we decided to compare two approaches to look at differential expression within subpopulations, one using pseudo-bulk approaches using HR, and the other one using mixed models as implemented by Muscat. This came from work from Mike Robinson's lab. There's a really nice paper published in 2020 that explores different approaches for differential state analysis. So first, I'm going to tell you a little bit about the results that we got with bulk RNA-seq. And what you have here, again, is the effect of sleep deprivation, a volcano plot. And we also did a treatment, a recovery sleep, to see how much a little nap can help reverse the effect. This is your PCA plot. We see a nice separation of treatment here. We have about 54,000 genes, and the sleep deprivation alters about, I would say, a third of the cortical transcription, and we only detect about 18,000 being expressed. And 79% is the number of positive controls from a microarray study that we're able to replicate. So we're really happy with that. We think this is very good levels of replication. We also we did a microarray study of the effects of recovery sleep. I didn't talk about that a little bit. So we had also positive controls for that. And we can see that 2 hours of a little nap makes you recover gene expression about 2,000 genes. And again, we have 72% or so recovery or positive controls. So we have a very large effect size. We are really confident that we know what we're seeing in typical conditions. So we move on to look at what happens in the mutant. And this is obviously a long story, and I'm going to make it really short. But if anybody's interested, you can look at our paper on eLife in 2019. What we can see is that the mutation enhances the effect of sleep deprivation on the regulation of gene expression. Overall, here is the number of genes that are differentially expressed between the mutant and the wild type. And their HC stands for home cage. They're just hanging out in their home cage. And after sleep deprivation, you double approximately the number of genes that are differentially expressed. And when you do a heat map of the differentially expressed genes, so these genes are all the genes contained in this Venn diagram, you can see that the majority of the genes are in this cluster that is mostly blue here and mostly red here. Blue means downregulated, red means upregulated, and you can see that in the mutants, this cluster of genes gets more blue or more downregulated. Now we are moving on to try to look at the same problem using single nuclear RNA-seq. And so we started with about 54,000 genes and almost 70,000 cells. Following quality control, we are left with about 60,000 cells. It's a pretty good quality data set. We did batch removal using the most variable genes. And what you're seeing here is just a t-SNE projection of all the 6 replicates together. So we have about 10,000 cells per individual biological replicate. And our first attempt of trying to assign cell types exclusively based on data wasn't that great. And why do I say that? So we collected positive controls that were obtained from in situ hybridization studies that were published of the mouse cortex. And we asked, how does the cluster assignment match the manual assignment of cell types? And this is because current best practice is try to combine computational manual assignment. And this is not a good assignment. And I'm going to walk you through why that is. So in the brain, you broadly expect your cell types to fall into 2 types, neuronal and non-neuronal. Within the non-neuronal, you have either glia, astrocytes, oligodendrocytes, and everything else, vasculature, blood, everything else that is in the blood. And within neurons, you have either excitatory or glutamatergic neurons or inhibitory or GABAergic neurons. So which means that the cluster here at the top should be either 3 or 4. And we have about 3. But this clustering is separating cells that will be together. So for example, here we have parvalbumin interneurons. These are GABAergic interneurons. And the other class of the most closely related is the somatostatin interneurons that are over here. So the correspondence wasn't that great. So we went on to try to look at annotation of the cell types. And what we see is that using the Allen mouse cortex SMART-seq levels, we get a much better correspondence of markers between, at least for inhibitory neurons and glia.
Lucia, we just lost your screen
However,
share.
when we go back and try to map those markers into the t-SNE projection, it doesn't look very good.
Lucia,
Yes?
we lost your screen share
Oh my God.
just a moment ago, only a few seconds ago.
Okay, let me do this again. Can you tell me where?
Just like the last slide, I think it was gone for about 10 seconds.
Okay. This slide or the one before?
Let's see the one before.
This one? Or this one?
Yes,
This
one. This one,
okay.
yes.
Yeah, this is so strange. I'm so sorry.
Yeah, no problem.
Let me go back.
And don't worry, if anything goes wrong, we'll be here in an instant to let you know.
Okay. So I was talking, let me repeat, about how our first attempt was to try to assign cell types using only clustering. And that was not very successful because it did not replicate very well the assignment based on our positive controls that we assembled from in situ hybridizations. So what we tried a different approach, which is to annotate cell types based on a reference data set. And disclosure, different references will give different labels. So you need to try different references and see how they perform. So what we ended up using is the Allen mouse cortex SMART-seq labels from the Yao et al. 2021 paper that were available. And here are our positive controls, and here is the grouping based on the annotation. And we see here that there's a really good correlation for this group of cells in red, and these are the inhibitory neurons. There's also a very good correlation for glia. We did not have any markers for non-glia non-neurons, so we don't expect any correlation here, but we don't have a very good correlation for excitatory neurons. On the other hand, that makes sense. I don't know if you notice, inhibitory neurons are named by their cell markers, where excitatory neurons are based on location. Right? So then we went ahead and we mapped the assignments that we got from the SMART-seq labels into the t-SNE projection, and it didn't look very nice. It looked very, very messy. And remember, this projection is all 6 replicates together. However, if we remove non-neuronal cell types, we actually get, and we also focus only on the cell types that have at least 500 nuclei, we get a much better clustering, right? So we use this cluster, and this is obviously very preliminary data, and this is work of Elena Zuin and David Risa's lab. We then looking at differential state transitions between that. And what I'm going to show you is the performance in one cell type. So this will be sleep deprivation versus control in glutamatergic neurons of layer 4, 5, IT cortex. And it doesn't really matter what this is because this looks very similar across all the cell types that we tested. What we see here is basically a volcano plot again, and a p-value distribution that you can see is very, very non-uniform. Upregulated genes are in blue, downregulated genes are in red, and what you have in this dark red are the positive controls for our microarray study. We don't expect to find them all because you lose sensitivity, and about 20% of them didn't replicate in RNA-seq, but it's a good data set to compare performance. So this is pseudobulk using HR, and this is the negative emollient mix model implemented through Muscat. And what you can see first is that the p-value distribution now looks much better and uniform. And you increase the detection level, and it looks like you increase the detection positive controls. So this is almost my last slide, which shows the performance of both HR or NBMM across all the excited the neuronal cell types that we are able to detect. And you definitely see that in all cases, using the negative emollient mix model increases detection and increases the detection of the positive controls. Now, there's a couple of very interesting things in this data that I just want to highlight in terms of biology. First, we have to really be careful about making these statements because power depends on how many cells you detect. But if you compare the two more abundant types of excitatory neurons with the two most abundant types of inhibitory neurons, you can see that sleep deprivation seems to affect a lot more excitatory neurons than inhibitory neurons. You know, with the caveat that you actually have to compare cell types that have similar numbers of cells. The other thing that I noticed right away is that there was one small subtype of inhibitory neurons in which the effect seems to be almost all downregulation. And I thought that was really interesting because what we had observed initially, both in microarray and RNA-seq, is that sleep deprivation in the cortex and in the hippocampus seems to downregulate a lot of genes. And then I asked, of all these differentially expressed genes that are downregulated, where can I find my eukaryotic initiation factors? And the answer is only in these cell types. So overall, this is the summary of the second part of my talk. By bulk RNA-seq, we are able to show that sleep deprivation has a large effect on the transcription that is actually relatively reversible with an app. We can replicate microarray findings really well while increasing the sensitivity and revealing new gene pathways that are influenced by the sleep loss. Using single nuclear RNA-seq, we can see that sleep deprivation seems to affect preferentially glutamatergic neurons, but also selectively downregulate transcription in a population of GABAergic neurons, including VIP neurons, and in particular, eukaryotic initiation factors. Why is this important? VIP, which is what VIP neurons make, is known to regulate sleep. VIP neurons are essential for circadian rhythmicity, and mTOR, protein synthesis regulation in VIP neurons, is known to regulate circadian clock synchrony and the stimulus sensitivity in the olfactory cortex. What are we doing right now? We are actually looking at the effect of sleep deprivation in our SHANK3 mutant mice. We have just finished collecting this data in the 10x multi-omic platform for both single nuclear RNA-seq and single nuclear ATAC-seq. But we have a lot of work to do in terms of data analysis. These are challenges that remain in the field. The biological variability in cell type assignment is huge. So what you're seeing here is our 6 mice. These are the home cage controls, and these are the sleep deprived, and this is all the different cell types color-coded. And I hope you can see how different each mouse is from each other. And the other issue that is important to take into account is we need to know how to model the different power for differential state analysis across subpopulations with different abundances. My final considerations for data analysis in general is that, confounders are always a problem regardless of the technology you use. They don't go away with newer technologies. The proper experimental design is always essential, but sometimes it may not be enough. It doesn't matter how much you try to control your experiment. These data sets are really noisy regardless. That's not an excuse for not trying to design the experiment properly, by the way. But the contribution of confounder is going to be different in different experiments. So you need to know your system, you need to know your controls, and you need to tailor the analysis pipeline to the data. So I want to thank the members of my lab. here, everybody has held a paintbrush, so everybody is involved in collecting the data, but the people I'm highlighting here are those that were responsible for a lot of the data analysis I showed you today. I want to thank David Arizo. He's been my collaborator and colleague for a very long time. A lot of the work that you saw were work by Davide and Dario and Elena. And I also want to thank the rest of my collaborators, my mentors, and my funding, and I'll be happy to take any questions.
All right. Thank you very much, Dr. Peixoto. Please, everybody, thank Dr. Peixoto for showing all of this cool, new, unpublished data and reminding all of us that just because it's cool and new doesn't mean that rigor doesn't matter. Thank you so much. If people have questions, it can be posted in the Q&A or in the chat. $35. Well, it looks like you left no doubt as to what was going on. Here we go. Did you use other clustering techniques such as UMAP or TISNI on the principal components, asks Bianca Granato from McGill.
If we're talking about the single nuclear RNA-seq, we use both TISNI or UMAP, and you get equally messy. So our TISNI plots are not that pretty. So what I showed you in the single nuclear RNA-seq holds true regardless of whether you use TISNI or UMAP, and is this problem of the cell type assignment. And it seems that the non-neuronal cell types are really contributing to the noise, right? And so what I showed you with TISNI also holds with UMAP.
No, Federico Marini asks, would a correction with RevSeq potentially be dangerous if, for example, it cleans up, quote unquote, too much of the data?
That is an excellent question, and the answer is yes. And this is why it's important to have controls. Right. So one of the things that I really glossed over is the fact that when you're using RUV-seq, you really need to do an iteratively exploration of how much variance you're removing. And you need to have a way to evaluate when it is too much and when it is too little, right? So in our case, we have independent, both positive and negative controls, which actually allows us, so we optimize RUV-seq to maximize the recovery of positive controls. When that is not the case and you're only using replicates, you have to be really careful. So I tend to recommend being conservative so that you don't clean up too much, right? But yeah, so that's how we do it. We use the positive controls from an independent experiment to basically decide where to stop.
Now we have an avalanche of questions. Maybe let's take somebody new who is not a BioSeq core member. Did you compare RUV-seq and SVA methods? Oh, this is a good one.
Yes, we did. So if you actually, so I also didn't talk about this, but on the 2015 NAR paper, we compare RUV so we compare a lot of different normalization methods, and also to peer. SVA and RUV-seq perform relatively the same, provided that you don't let SVA estimate the number of Ks from your data, and you actually do this iterative process, right? So that is it. But that data and that comparison is in the 2015 NAR paper. If you want to take a look at it, SVA performs equally well. That just don't let it estimate the variance to remove. You have to go through this exploratory data analysis of how this is working.
Then Rohit Satyam asks, are your pipeline available for bulk RNA-seq, which sounds like an invitation to pitch the nucleic acids research paper again.
Yes. So that nucleic acid research paper that, as Tim just mentioned, has a tutorial that has the pipeline and actually allows you to replicate all the figures. That's a tutorial that Davide put together. And it also walks you through a very important issue, which is I see is the next question up the line, which is how do you do when you don't have controls, and how do you know where to stop, right? So one of the things that we actually showed in that paper and that tutorial is that if you don't have negative controls, housekeeping genes do a very good job. Actually, this approach is pretty robust to the negative control set. It is not as robust to not having replicates. So having biological replicates and being able to estimate the variance between the replicates is really important. Right? But yes, if you want to take a look at that paper, you can look at the tutorial. You should be able to replicate all the figures. And if you cannot, tell me.
So we have a question from Thady Moreno at UCSF. What happens in the case of not having any type of replicates, like is the case from tumor patient samples?
So that is a really good question, and I don't know. So that is actually one of the cases in which maybe RUV-seq will not help you very much. That's why I like having mice and being able to make as many replicates as I want.
Then Tiago Silva asks, can you share the source of the clusters marking for hippocampus that you're using? Which ones are markers for excitatory neurons, inhibitory neurons, and so forth?
Just one thing for the single nuclear RNA-seq, that is cortex. And I'm not sure if you're referring to the markers. So the SMART-seq assignments were taken from that paper, the Yao et al., Cell 2021. The markers for in situ, I put them together myself, and hopefully when we'll have them out soon in the preprint. But I can potentially, if you email me, I can send you I use reviews of in situ. I can tell you the sources that I used, right? But they're both available. So if you're interested in the in situ markers, email me. Otherwise, we use the SMART-seq from the Yao et al. Cell 2021. And this is part of the Allen Cell Census Network. And so all their data is public.
Thomas Schauer from LMU asks, did you investigate the source of unwanted variability? Batch effects, mouse litter, library prep, blocked versus completely random design?
So initially on the first microarray data set that I show you, the fear conditioning one, I did a lot of that. So I actually did that even before I collected the data, because when I arrived at the lab, I had a lot of data sets that never made it to light because they had batch effects. So the reason that I designed the experiment the way I did was to correct for all the things I know happen. I can tell you all of those things can include give unwanted variability. They were not an effect in our design because we control for that. In the microarray data set, the first two principal components were actually smell and dominance. So we switched then from basically hosting the experimental mice away from the colony. So we could actually map in the initial ones. These mice were all males. So depending on how many females they had around, that actually produced a really strong effect on gene expression. Right? So we mapped that to that. But the bottom line is that if you randomize this properly, it doesn't matter what the confounder is as long as you can remove it, right? But the other thing that I didn't say, mouse litter is really important. So when we do our designs, we're always using brothers compared to the mutants, and we always randomize litter. We always prep the libraries together. So anything that I could see from the previous microarrays, we controlled, and we still see unwanted variability. Time of day is the other important one. So that I didn't mention. The reason that we did it over 9 days is you want to be within 2 hours, or time of day becomes a very strong unwanted effect on your gene expression pattern. So not that anybody ever reports which time of day actually are taking their samples, but time of day together with sleep status, that's the other thing. If you can appreciate by the second, depending on which time of day you're taking your samples from a mouse, the mouse will be sleeping more or less or not. And that is also going to change your gene expression effects.
All right, well, it looks like we're at the top of the hour. Thank you again, Dr. Peixoto, for a wonderful presentation and for answering a great many questions. Hope everybody has time to stew over them and think about how this applies to your own research and enjoys the rest of the conference with this in mind.
Thank you, everybody.